Disintegration test. This test determines whether dosage forms such as tablets, capsules, suppositories disintegrate within the prescribed time when placed in a liquid medium under the prescribed experimental conditions.
Disintegration is considered when A. No residue remains on the screen. B. If it is residue, it consists of mass having no palpably firm, unmoistened core. C. Only fragments of coating for tablets and shell for capsules remain on a screen. D. If disc-used fragments may adhere to the lower surface of disc.
DT apparatus node. Use apparatus A for tablets and capsules that are not more than 18 mm long. For larger tablets and capsules, use apparatus B. Basket rack assembly. As per USP Chapter 701, tubes for apparatus A is 6 and for apparatus B is 3 numbers. Length of tubes for both apparatus is 77.5 plus or minus 2.5 mm. Inside diameter for apparatus 20.7 to 23 mm and for B 32 to 34.6 mm. Wall thickness is 1 to 2.8 mm for apparatus and 2 to 3 mm for B apparatus. Number of plate for both apparatus is 2. Plate diameter is 88 to 92 mm for apparatus and 95 to 99 mm for B apparatus. Plate thickness 5 to 8.5 mm for apparatus and 7.5 to 10.5 mm for B apparatus. Number of plate hole for apparatus is 6 and for B apparatus is 3.
Hole diameter range for apparatus A 22 to 26, for apparatus B 33 to 34 mm. Wire weave gap range 1.8 to 2.2 mm. Wire diameter for apparatus A 0.57 to 66 mm and for apparatus B 0.025 inch. Disc thickness for apparatus A 9.5 plus or minus 0.15 mm and for apparatus B 15.3 plus or minus 0.15 mm. Disc diameter for apparatus A 20.7 plus or minus 0.15 and for apparatus B 31.4 plus or minus 0.13. Specific gravity range for both apparatus A and B is 1.18 to 1.20. Number of holes 5 and 7 respectively. Hole diameter for apparatus A 2 plus or minus 0.1 and for apparatus B 3.15 plus or minus 0.1.
DT apparatus having control unit, stroke length pointer, temperature sensor, basket, basket holding arm, water bath.
Criteria for uncoated or plain coated tablets. 1. Place one dosage unit in each of the 6 tubes of the basket rack assembly and, if prescribed, add a disc. 2. Operate the apparatus using water or the specified medium as immersion fluid maintained at 37 plus or minus 2 degrees Celsius. 3. At the end of the time limit specified in the monograph, lift the basket rack assembly from the fluid and observe the tablets. All the tablets should have disintegrated completely. If 1 or 2 tablets fail to disintegrate completely, repeat the test on 12 additional tablets. If 6 tablets are tested, all 6 of the tablets are disintegrated. If 18 tablets are tested, then 16 out of 18 tablets are disintegrated. Criteria for delayed release tablets and capsules: acid resistant or enteric coating. 1. Place one dosage unit in each of the 6 tubes of the basket. If the dosage units are not sugar coated, proceed to the acid stage. If testing tablets that have a soluble external sugar coating, immerse the basket in water at room temperature for 5 minutes and then immediately proceed to the acid stage. 2. If specified in monograph, add one disc to each tube. Acid stage: immersion fluid, 0.1 molar hydrochloric acid, or simulated gastric fluid as, or is specified in the monograph. Temperature: 37 plus or minus 2 degrees Celsius. Evidence of disintegration, cracking, softening. Proceed with the buffer stage. Buffer stage: immersion fluid, pH 6.8 phosphate buffer, or simulated intestinal fluid as, or is specified in the monograph. Temperature: 37 plus or minus 2 degrees Celsius. Time, as specified in monograph.
Criteria for effervescent tablets: place one tablet in each of 6 beakers containing 200 milliliters of water. A suitable beaker will have a nominal volume of 250 to 400 milliliters. Time, 5 minutes or as specified in monograph. Evolution of gas around each tablet or its fragments has ceased. Each tablet has dissolved or disintegrated and has been dispersed in the water so that no appreciable agglomerates remain.
Number of cycles per minute: the number of cycle per minute is within 28 to 32 thought the period of operation. Calibration of DT apparatus. Calibration of distance traveled using a calibrated ruler, pen, paper. Distance traveled is between 53 to 57 millimeters. Temperature verification using calibrated thermometer. Temperature remains 37 plus or minus 2 throughout the operation. Count number of cycles up to 5 minutes.
DT for suppositories apparatus. The apparatus consists of a sleeve of glass or suitable transparent plastic of appropriate thickness, to the interior of which is attached by means of 3 hooks a metal device consisting of 2 perforated stainless metal discs each containing 39 holes 4 mm in diameter. The discs are about 30 mm apart. The test is carried out using 3 such apparatus each containing a single sample.
DT for suppositories. Each apparatus is placed in beaker with capacity of 4 liters water. At 36 to 37 degrees Celsius. Apparatus together placed in vessel with capacity of 12 liters. Beaker is fitted with slow stirrer and device which holds cylinders vertically not less than 90 mm below the surface of water and allow them to be inverted without emerging from water. Use 3 suppositories. Place each one of the lower disc of device. Place the latter in the sleeve and secure. Invert the apparatus every 10 minutes. Examine sample after the period prescribed in monograph. Acceptance criteria for suppositories. For molded suppositories, DT occurs is not more than 30 minutes. For fat-based suppositories, not more than 60 minutes. Uncoated tablet 15 minutes, film-coated tablets 30 minutes, sugar-coated tablets 60 minutes, dispersible tablets 3 minutes, effervescent tablets 5 minutes, enteric-coated tablets 2 hours in HCl no disintegrate, 60 minutes in buffer disintegrate, soluble tablets 3 minutes. Uncoated tablets 15 minutes as per IP, BP and 5 minutes as per USP. Sugar-coated tablets 60 minutes as per IP, 30 minutes as per BP not applicable as per USP. Film-coated tablets 30 minutes as per IP and BP not applicable as per USP. Enteric-coated tablets 1 hour as per IP, 1 to 2 hours as per BP, 2 hours as per USP. Effervescent tablet or dispersible tablets not more than 3 minutes as per IP, BP, USP. Or a dispersible tablet not more than 1 minute as per IP, BP, USP. DT is not for buccal, sublingual and chewable tablets, because they are directly absorbed. No DT for sustained release and controlled release. Difference between disintegration time, DT, and dissolution in pharmaceutical testing. 1. Definition: time taken for a tablet or capsule to break into pieces. Measure of the rate and extent of drug release in solution. 2. Purpose: checks if the dosage form breaks down properly, evaluates how much and how fast the drug dissolves. 3. Test medium: usually water or buffer, simulated gastric or intestinal fluid. 4. Endpoint: tablet breaks apart, no firm mass remains, specific % of drug dissolved over time. 5. Instrumentation: disintegration tester, basket rack assembly, dissolution tester, paddle, basket apparatus. 6. Importance: precursor to dissolution, critical for bioavailability and therapeutic effect.
Disintegrating agents: 1. Starch. 2. Sodium starch glycolate. 3. Crosscarmellose sodium. 4. Crosspovidone, cross-linked PVP. 5. Microcrystalline cellulose. 6. Alginic acid and sodium alginate. Parameters effect on DT: 1. Type and concentration of disintegrants. 2. Binder type and amount. 3. Lubricant type and amount. 4. Filler, diluent properties. 5. Tablet hardness and porosity. 6. Compression force. 7. Granulation method. 8. Moisture content. 9. Medium pH and composition. 10. Storage conditions.
Reference guidelines. 1. USP chapters 701 and 2040. 2. European Pharmacopoeia 5. 001 2005. 20902. 3. ICHQ 4B Annexure 5 R1. 4. JP 6.09 Disintegration Test.